Okay, so it's 9 o'clock. Let's get started. Hello, everyone. Welcome to our today's Amy Zoom seminar. It's really my pleasure to welcome our today's speaker, Dr. Mitchell Hove, also my colleague from NCI as a senior investigator. As I mentioned, Mitchell currently is senior investigator in laboratory of molecular biology in NCI NIH. He got his PhD in immunology from University of Illinois at Urbana-Champaign, and then as Ruth Kristen National Research Service Award postdoc fellow. And then he joined NCI for a postdoc training, and then was appointed as a tenure track investigator in 2008, start his independent research program. Now, after 7 years, 2015, he was promoted to senior investigator, and then currently he served as a deputy chief of the lab in molecular biology and director of antibody engineering program at NCI. So Michel, his lab conducts research in biochemistry and molecular biology with a focus on glypican biology, antibody protein engineering, and cancer immunology, with a primary interest in the machinery of cell surface receptors. His team made really uh, groundbreaking contributions to understanding the biochemical and immunological properties of glypicans, including GPC1, GPC2, and GPC3 in cancer development and immunotherapy. His lab has advanced the knowledge of glypicans in Wnt signaling and YAP signaling, their structure function relationships, and the discovery of previously unidentified associated proteins such as FAT1 and AFP. His lab also developed antibody and T-cell based immunotherapy for the treatment of liver cancer, pediatric cancers, and other malignancies. Now, in addition, his lab developed antibody engineering tools such as mammalian cell surface display technology and Shark camel and nano bodies libraries to advance this drug discovery. Some of their research had direct clinical applications. Now, as I mentioned, Mitchell is the founding editor in chief of Antibody Therapy and served on the boards of directors of Antibody Society and the Foundation for Advanced Education in the Science. He received the APAO Scientific Achievement Awards, uh, Frank's Francis Awards for Mentorship Excellence at HHS, and the NIH Innovation Awards, and the NCI Director Innovation Awards. He was elected as Fellow of the American Institute for Medical and Biological Engineering in 2023 and named the Asian American Engineer of the Year in 2024. So really excited to have Mitchell with us today. And then I think today he's going to tell us about his latest work on engineering CAR T cells to target solid tumors. So with that, thank you so much, Mitchell, with us today. And then we are very much looking forward to your talk.
Thank you, Dr. Wu, for very kind introduction. Thank you, everybody, for joining the seminars. I heard Dr. Wu and Dr. Huang told me that the many students may be joining for the seminar series. And feel free to ask questions at the end of my talk. And very happy to answer any question, particularly from junior investigators, junior faculties, and students. We always encourage the student to ask the question first. And that's what I want to say. Okay, thank you very much. And today I'm going to talk about the engineering CAR T cells to target the solid tumors. And this is my disclosure. It just shows, just says what the content of my lectures represent my own views, not NIH or US Department of Health and Human Services. And I own the patents and assigned to the NIH. And I also received the research fund from pharmaceutical companies as listed here. So in terms of antibody therapy, I recently wrote a paper with John Hopkins group, and we co-authored this paper in Nature Reviews in Cancer. And in this paper, we described what antibody therapy is about. So as you can see, the antibody is like this. Is like in your textbook, it's IgG kind of format, and it's a Y shape, which have heavy chain and light chains. So when antibody bind to the tumor-specific antigen like HER2 on breast cancer, it could kill the cancer cell directly, just antibody alone, either engage the NK cells or macrophage, and also could be also engage the complements. And the antibody could also inhibit checkpoint inhibitors, as shown here, PD-1 and PD-L1. So many tumors has PD-L1 expression, and the T cell has PD-1. So antibody could block this kind of interaction. Obviously, PD-1, PD-L1 is one of the many checkpoint interactions, and many of them probably still unknown. And antibody can be used to make a so-called bispecific antibody. In one approach, the antibody could target the tumor cells to engage the T cells, anti-CD3, and on one arm, and anti-CD20 on leukemia cells on the other arm. In this way, the T cell, so-called T cell engager, could kill the tumor cells. On the other hand, it could be bispecific antibody targeting simultaneously two antigens. One is EGFR, the other the MET. And by doing this, synergistically induce the cancer cell death. That's very elegant mechanisms. So, but the antibody could also use as a guide to guide the cytotoxic drugs, like small molecules. And could be radioisotope, and it could be protein toxins. And in this case, I'm personally involved and for one of the FDA-approved drugs. And so for the ADC, the antibody and the immunotoxin and the radiopharmaceutical drugs basically targets the tumor-specific antigen, internalizing the tumor cells. So obviously it needs to be internalized. Not all the antigen can be internalized. For example, CD19 is not internalized, and the CD20 or 22 could be internalized. That's why you don't see here CD19 ADC, because it's poorly internalized. But anyway, once it's internalized, you can kill the tumor cells with various mechanisms, whether it's inhibit the mitochondrial or mitosis, and it could be DNA damaging agent. So there are several mechanisms you could do. And my team and the NCI group, we actually developed the anti-CD22 immunotoxin led by Dr. Ara Paston, and he was my postdoc mentor many years ago. And we developed this FDA first approved immunotoxin, and still the only drug in this category, and it was developed in NCI and eventually approved by FDA. So it was really very rewarding to have this immunotoxin targeted drug-resistant leukemia or lymphoma. And in this same review, we actually looked at all the solid tumor, in addition to the leukemia and the B-cell hematological cancer. You can see many solid tumor. There are not many targets. Okay, so for example, pancreatic cancer, there are no targets, FDA-approved drugs. Same as prostate cancer, same as brain tumor, and many other cancers. Even in later stage of clinical trials, there's some repurposing HER2 as antigen in pancreas, but not really pancreas-specific antigen. And then we have B7-H3 and others in liver cancer, but really, and repurpose HER2 in liver cancer, sorry, and B7-H3 in prostate cancer, and so on. So ovarian cancer, and we have very long-standing CA125, but other than that, it's quite a few. So I guess everybody will be very aware of that. Tumor-specific targets are very rare, okay? And new tumor targets are needed. So in my lab, as Dr. Wu introduced in the beginning, we have been studying GPC3 in liver cancer, and more recently, GPC2 in pediatric cancers, and GPC1 in pancreatic cancers. So I want to focus on that in today's talk. I try to explain what we did. Since, as I showed in the title, so I want to talk about CAR-T today. So what a CAR T is? So CAR T basically is reprogrammed T cells using antibody to lead the T cell to target the tumor cells specific to the tumor specific antigens. So this is really one of the first example for synthetic biology. And T cell, CAR T cells is really synthetic T cells. So it need a lot of engineering on the T cell side using antibody and many other co-stimulatory factor and T cell signaling molecule to make it more potent and to be able to guide it to the tumor cells. But the CAR T cells for solid tumor is a challenge because in order to get rid of the solid tumors, the CAR T cell need to overcome the extracellular barrier and need to get into the intracellular, intratumor environment, tumor microenvironment, we usually call. And they have tumor-associated myeloid cells, they have Treg, which is the suppressive immune cells. And they also have immunosuppressive molecules like TGF-beta and IL-10. And on the other hand, as I said, the tumor antigen really are the key, and there are not many tumor antigens. So my lab has been focused on glypicans as tumor-specific targets. So the goal in my program is to use antibody engineering to develop a drug for solid tumors. So as I described, we use ADC, we use immunotoxin, we use T-cell engager, we use CAR-T and K. So every single molecule I listed here, we have a person in the lab working on it right now. So for today's, I'm mainly just focused on the CAR-T. And in addition to glypicans, we also looked at other tumor antigens such as mesothelin and B7H3. I probably don't have time to talk about mesothelin today. For glypicans, there are 6 in the members in the protein family. In this family, there are GPC1 to GPC2 and GPC3 all the way to 6. They all have a common feature, that is the protein, and with a GPR anchored on the cell surface. They have heparan sulfate chain, which is negative charged and a very sophisticated heparan sulfate chain. We spend a lot of time trying to analyze that. And they're the core proteins. So some of the member has more than 2. GPC3 has only 2, but like GPC3, like a sugar ball, they have 5 glypicans. You can imagine when we make an antibody, one of the first barrier we deal with in this protein family is they have too many glycosylations. And it's not immunogenic. When you immunize in the mice do not respond because they're covered by sugar. And just like a heparin, people use, so patients could use the heparin isolate from pigs, from ox. There's no immunogenicity in humans, right? So the heparin is used in the clinical ER and the ICU, no issue of immunogenicity. That is true for heparan sulfate because they are really even immunoinhibitor. So one of the first things we want to deal with is how we can make antibody for those things. And why we interested in glypican? Because oncofetal antigen are present in fetal development and silencing adult normal tissue expressed in certain cancers. So that means in normal adult tissue, usually they silenced. They are only in early development, like a fetal liver, they have GPC3. But an adult liver does not have GPC3 expression. But unless you have the liver cancer, it's upregulated again. And so we hypothesize those membrane protein is oncofetal antigen may be suitable for antibody and cell-based therapy. And over many years, we actually studied this mechanistically, how GPC promote the tumor growth. One of the mechanisms we found, as Dr. Wu introduced, we also studied the YAP signaling, but I want to focus on Wnt here. And we found that GPC3 attract the Wnt. It's like a concentrator and also a transporter. So they concentrate the Wnt3, particular Wnt molecule, and concentrate and transport to the phrasal, and therefore, and the beta-catenin signaling elevated, lead to upregulation of some Wnt targeted genes we actually study in HCC. And biochemistry, from biochemistry point of view, we actually look at the GPC3, how it binds to the Wnt. Well, we find surprisingly, which is not intuitively in the beginning, makes sense. that is GP3 bind the protein core and the heparin sulfate chain. They need to bind both, not only just one of them. And here's the model we just published a couple, actually last month. And so in this invited review paper, we summarize what we know and what other people have been done so far. And the GPC3 actually bind to the Wnt. And we mutated F41, for example, here in one of our hepatology paper, we found that if we mutated this phenylalanine 41, and you lost the Wnt binding to GPC3. And so the Wnt3 bind to the frisol, which is GPC receptors. That's a canonical complex. And the GP3 bind on the other side of Wnt3 and form a really nice complex here. And all of them together is a super signaling complex on the tumor cells. And GPC3 expressed in fetal liver, placenta, but not in normal liver. So, but it's highly elevated in 70% of hepatocellular carcinoma. That's one of the major, actually the major primary liver cancer. And we develop antibody widely used now. It's called YP7, and it has been developed for ADC, T-cell engager, CAR-NK, CAR-T cells. So Centibio just initiated CAR-NK using our humanized YP7 for treating liver cancer in 2024. Actually, the trial is running in China right now. And so in my lab, we published a series paper using this same antibody to do ADC or CAR-T or T-cell engagers. And you can see this antibody binds HCC tumor, but not any normal livers. And it does not bind any normal organs like pancreas, stomach, heart, kidney, nerve, and so on. And using this antibody, several years ago, we actually developed CAR-T cells. That's one of the first CAR-T cells we developed in the lab. And we use VH is a heavy chain variable region with linked to the light chain variable region and form a single chain FV, humanized YP7. So we humanize it using antibody engineering to make it more like a human antibody. And then we connect to CD8 hinge and CD8 transmembrane domain with 41BB co-stimulate domain and the CD3 TCR signaling. And we also have a human EGFR truncated form. It's not a functional EGFR. It's just the epitope recognized by cetuximab, which is FDA approved drug. So we could remove the CAR-T cells if we no longer need it in patients. So we published this paper in gastroenterology in 2020, and during the pandemic in January 13, 2022, we started treating the first patients in NIH clinical center. And so this is ongoing. And we also interested as a molecular biologist and immunologist, we always interested in mechanism, why CAR-T cell works. And so we actually get a CAR-T cell from mice and do single cell suspension, incubated with tumor cells to see what actually CAR-T cells are functional and why they are functional. So while we're doing that, we find actually it's only 0.5 to 1% of CAR-T cell among all CAR-T cells are polyfunctional, meaning so only very few CAR-T cells, such as here, and the same T cells producing gamma interferon, perforin, TNF-alpha, and so not every single CAR-T cells producing multiple cytolytic factors, only 0.5 to 1%. So we call it polyfunctional CAR-T cells. And so we believe those so-called polyfunctional CAR-T cells are actually the major players in the CAR-T cell therapies. And we further look at those polyfunctional CAR-T cells, we call it, and we do the genome sequence of all the pool of CAR-T cells in the tumor microenvironment. And we find that in different mice, in different animals, some of the CAR integration site is enriched if you do T-cell genome sequencing. And those integration sites, for example, we found NUPR1 is a nuclear protein, apparently enriching the CAR-T cells from the 5 weeks to 7 weeks, and it's highly enriched. But before the treatment, in the early treatment, is not enriched, but later on, it's enriched. And we don't see any enrichment when in the control CAR-T cells, obviously. And we do this not only for GPC3, we also do GPC2 and B7H3. we try to find those common hotspots. And we think maybe if we know those hotspots, we might be able to engineer polyfunctional CAR T cells in vitro before we put into patients. So instead of we put 100 million CAR T cell into one patient, we probably only need several hundred CAR T cells, maybe even need just one CAR T cell, because it probably is only one or two very potent CAR T cells do all the job. If it's lucky, that CAR T cell and expand and kill the tumors in vivo, because these are live drug. It's not like any other drug. They expand, they grow in patients and kill tumor cells whenever tumor cells are. And the CAR T is very exciting because that's really one of the first drug besides surgery. CAR T is probably only one of the first such drug. They can kill very big tumor. And because they are live, they can keep killing until the last tumor cell is dead. So if it works, it could work really, very well. Okay, we also do some single cell RNA-seq in polyfunctional CAR T cell. They said I'm not going to very detail because we looked at which gene is upregulated or downregulating the high polyfunctional CAR T cell, but not the low polyfunctional CAR T cell. We are again using the CAR T cell from the animals. And we are using the same approach going to analyze the CAR T cell in patients for ongoing clinical trials. So hopefully we learn more after that. And beyond the GPC3, and in probably 5-6 years, actually it's more than 6, now 7 years, and we actually start to look the other glycans. And one of the first things we work is called GPC1. So GPC1 was reported before as exosomal markers for pancreatic cancer, indicating maybe it's a good marker. But we don't know whether it can be used as a therapeutic target. So we look at all the pancreatic cancer cells. We find that GPC1 is only expressed in pancreatic cancer cells, but not in normal pancreas. Okay, and we have, and if like we do in the GPC3, if we put the report assay, okay, into the put the GPC1 in the report assay, we will see the GPC1 is also upregulated the Wnt signal. Just like GPC3 we use here as a control because we already know this more than 10 years ago. Okay, and if we knock out the GPC1, and on the Western blot, you can see the active beta-catenin is significantly downregulated. And in 3 different clones, those 3 different clones we sequence confirm they are actually independent, different individual clones. And all of them, if we knock out the GPC1, you will see the downregulated active beta-catenins. So that indicating, like a GPC3, GPC1 is also regulated Wnt, but in different cancer type. In this case, it's pancreatic cancers. And to isolate the monoclonal antibody against GPC1, we took 2 approaches. In one approach, we immunized black 6 mice and to make a hybridoma and fusion, and with masking code method, and to screen the fusion and find the antibody called HN2. In the other method, we actually create a camophage library in the lab. It's called VHH, heavy chain only antibody. Using this one, we pan on the GPC1, we find an antibody called D4. And D4 is a nanobody. Well, the reason we like nanobody is because they tend to bind and bury the functional site because they are small. And the protein surface is not flat. And they always have a cavity on the groove. And those groove on the cavity, sometimes we don't know what the purpose is. Normally, that's where ligand or receptor tend to bind. And the nanobody tend to bind those, otherwise it will be washed away during the screening. So the beauty of nanobody, they tend to bind those functional sites, and they are small. It could actually induce very good transduction efficiency, and also they cross mouse and human, so we can use mouse tumor and mouse tumor models. So like immunocompetent mice. And as you can see, the nanobody here, it binds to this side of the GPC1, and the Fab is bound to the C-terminal end of GPC1. And so as I said the nanobody could actually go to the cavity of the surface protein. Unlike a normal conventional by VHVL, their surface is relatively flat. Okay? And nanobody could bind to the cavity does not mean they could not bind to the surface like this. Nanobody, in order for nanobody to bind a flat interface like the one on the left, the nanobody need to orientate it differently. So normally nanobody have to be on the side, like this side, to bind to interface like this. So nanobody could still, we have example like that. Nanobody, once they oriented there differently, they could still bind a flat interface. But normally, if you engage the so-called CDI regions, a nanobody usually go to the cavity. And this is so we made a camel. It's actually it's a one hump camel, and so it's an Arabian camel, and so we can use those camels. And so we take the blood, camel blood, and PCR amplify the nanobody genes and put in the phage. That's a bacterial virus basically, and so put into a bacteria, they amplified for crazy. And so it would be 10 to the 11th diversity, and each of the library. So we make each library from each camel. So for 20 camel, we have 20 library. In reality, when we screen, we put them together, and then we can pan the nanobody. So when we find this nanobody, when we plug it into our car, we found a common problem. So if you're replacing single chain V with just nanobody in a conventional car, you immediately have some issues about the folding and the stability. So because it's just probably it's not the flexibility of the hinge. So we need to actually shorten the hinge. Instead of use 45 amino acid hinge, we actually shorten. We try all different lengths of hinge in our paper. But ultimately, we find the 12 amino acid hinge works best for the nanobody. So then they were actually for very nice dimers for the CAR-T. And using that, and we have IgG4 hinge, which is a shorter hinge, versus CD8 hinge, which is a long hinge, 45 amino acid hinge. The short hinge does work very well. This is the pancreatic cancer models, and you can see it's actually efficacy is improved if we shorten the hinge. So in that case, we do some kind of engineering on the nanobody-based CAR to make the hinge short to be more actually compatible for nanobody to put into the CAR format. And besides the GPC2, GPC1, we also start to work on GPC2. We found that GPC2 is targeting childhood cancer, particularly neuroblastoma we focus on. And we were among the first actually published the paper report that GPC2 is an oncofetal engine expressed neuroblastoma, and also independently confirmed by UPenn group in the same year later. And we found the GPC2 knockout could reduce NMEC expression, which is a key oncogenic driver of neuroblastoma and downstream target of Wnt signaling. That is quite interesting. And that is already published in that PNS paper, so I'm not going to talk about that. And in the last couple of years, we actually look particularly at isoform. And we found that GPC2 has a tumor-specific isoform called 201. So that isoform has extra exon 3, exon 10, exon 7, 8, 9. So our hypothesis is if we target exon 3, for example, we could target only tumor, not any potential normal tissues, okay? Because they don't have, normal tissue does not have these exons. And if you look here, all the normal tissue, even at the RNA level, it does not have this exon 3 except testis. So that means testis is the something you will be, if you talk about side effect, testis will be the one targeted by this anti-GP2 CAR-T. But in general, testis are already destroyed during the chemotherapy and the radiotherapy. So I think that's something we need to take care of, but it's not something you can do with it. And so we isolate the antibody called CT3, and it indeed it binds exon 2. And as you can see, the CT3 Fab actually integrate exon 3 and also exon 10. Both are only present in the tumor-specific isoform, not the other isoform normally expressed in the normal tissues. And again, as we predicted, if we do tissue staining on all the normal tissues, it's quite clean, except that this antibody still bind testis, which is not surprising to us, because on the RNA level, we see it. And using this CT3-based CAR T cells, we could regress the neuroblast. This is a very tough neuroblastoma model. So we grow them usually 4 weeks, and the tumor cell need to spread, settle, and seed and grow, and take a while. And then when it takes about a month to grow, and then we start to treat it. And those mice are really miserable because they have tumor from the skull all the way along the spinal cord, then go to the leg. So it's really mimic what's going on the neuroblastoma in human. It's a metastasis to everywhere where neuron is. They just go around the neuron. Wherever there's the neuron, there's a tumor there. It's a neuroblastoma. And we could actually get totally tumor-free using these CAR T cells. And all might survive. So because of this really promising data, we're actually going to run a trial later this year, maybe early next year, because of the GMP production. It's a little bit delayed right now, but I think we are already past it. And the NCI already approved the protocol. It's just waiting for FDA to submit the IND and get approved. So this is going on, going to happen very soon. And another company working with us called CRISPR Therapeutics, they published in SITC meeting recently. They use this CAR for allogeneic CAR T cells, off-shelf this CAR cell therapy. That is also going on. And in the last, I think 5 minutes or even 10 minutes, I think, so I can discuss something which is unpublished, but and we are actually still interested in doing that. That is the success of the CAR T cell therapy is in hematological cancer is partly due to the abundance of antigen like CD19. So when we talk about CD19, why CD19 is so successful? One of the many reasons, of course, that's not the only one reason. One of the many reasons is the CD19 has a really abundant size. So you have 10,000 or even more, 50,000 molecules per cell. But one of the challenges for CAR T cell facing solid tumor, besides the physical barrier, immunosuppressive cells, immunosuppressive molecules, is the antigen density is not compatible. We are talking about the antigen density. They are tumor specific, but they are probably only 500 molecules or even just 200 molecules per cell. So we heard it's a heterogeneity of the solid tumor. That is because a lot of solid tumor, the antigen density is very low. So, but for tumor-specific antigen like GP2, GP3, not the EGFR, it's basically absent in normal tissues. We could improve the CAR activity significantly, not worrying about on-target off-tumor toxicity. So that's the advantage. So what we try to do is make a CAR more active. So one thing we do is, this is the current CAR, and the new CAR we are doing is using the GP2 Fab gamma delta TCR. So we put the gamma delta TCR format here and fuse with a Fab of antibody. So by doing this, we call AB TCR. Its reality is a Fab TCR. And we do this, it's more like a TCR receptor. So if you look at the CAR, it's not really like a T-cell receptor. It's a very artificial molecule. And the reason we believe the CAR might be not so potent for solid tumor, there are many reasons. One of them is constitutively active makes the TCR CAR T very exhausting. That's one of the reasons. So may reduce persistence. The other thing is it's not really a receptor. It's not how T-cell receptor looks like. It's more like this, the T-cell receptor. So they are non-covalently recruit the CD3 signaling molecule endogenously in T cell. And unlike here, you covalently link to the CD3 zeta in the traditional CAR. So we try to say why we do not make a CAR just like TCR. So that's how we did it. And then, of course, we also need a co-stimulation factor like a CAR, but we put a co-stimulation factor separately, not covalently bound in one molecule. So in that way, we are more mimic what's going on TCR in the T cells. So by doing this, we start to test our hypothesis whether we can treat cancer cell with a low antigen density. So obviously, some of the cancer cell line we use are very low density, like this, LAN1, I'm sorry, and this one is also low. The tumor cell line I show you in the previous slide, we show very beautiful activity. It's MI5. It's also low, but not that low. It's like 2,000 size per cell. Consider leukemia, lymphoma, like CD19, CD22, CD20. This is much, much lower than that. But still, it's higher than this one. It's only less than 500 size per cell. So by doing this, we compare FTCR with CAR, and FTCR is really potent. Okay, it can shrink the tumors. CAR still work, I mean, very impressively, right? But just not as good as FTCR. And so because of that, we also, obviously, like any scientific experiment, we need to repeat many times. We also want to try second model. So in second model, we recently done, in just only last couple of weeks, we actually do this model. We can see also, and in this case, we do isotopic model, and it shows very potent again. And when we look at the mechanism, one of the mechanisms we see very obviously is the T cell, infiltrate T cell in the FTCR group is a much, much higher number in consider the conventional CAR. Obviously, the control is CD19. It does not bind the solid tumors. And so the green is T cells. That's CAR T cells. And so, you can see that in FTCR, you can have a much more infiltrated CAR T cells in the tumor microenvironment. And obviously, GP2 label the T cell, tumor cells. And the tumor cells just reduce in the FTCR because it's the efficacy. And this is somewhere around here, like a day 10, even before probably day 9 or day 8. Yeah. Okay, so we also see, I did not show the data here, and FTCR could enhance stemness of T cell by enrich the many memory markers you can imagine. And so we jointly find pattern with Eureka through NCI, and Eureka will sponsor clinical trial at the NIH as well. So we hope that's going to happen early 2026. So the major finding in my talk I want to summarize today is I tried to tell you GPC1, GP2, GP3 are new targets for treating pancreatic cancer, pediatric cancer, and liver cancer. And I think tumor-specific isoform are very exciting area of research, and we are very excited about this. Identifying tumor-specific isoform is very promising for discovering new targets, as we show in GPC2. We also find other isoform in other targets, and that obviously is a very attractive approach. And we find that TCR-like CAR is more mimic-like TCR rather than CAR, is more active than CAR in particular low antigen density tumors with better tumor T cell infiltration, persistent expansion in the tumor microenvironment. And as I told you, we have one ongoing clinical trial in clinical center targeting GPC3 for liver cancer. We also have 3, they all developed in my lab, and we get, actually I got a funding from NCI and NIH, and some with companies. So we actually running a trials either this year or early next year for GP2 and trials. And we also have mesothelioma I don't have time to talk about. And we also developed this CAR. It's called humanized YP208. Yeah, that is targeting. So we are recruiting patients for this trial already. So we are screening patients right now. So with that, I think thank you everybody. I'm looking forward to answering any question you may have. Obviously, I have very fantastic student fellows in the lab over the years. I can't list them all because there are many former members. I also have current collaborators, and we get money, funds from NIH intramural research program. We also get funds from a company I mentioned, and they actually help us to support some basic research and translate into the patient care. And with that, so thank you very much.